Pick up any biology textbook. Open it to the section on plants. It'll be right there, written with the confidence of an undisputed fact. Plants and bacteria are fundamentally different life forms. They do not exchange genetic tools. What if I told you there's a common garden shrub found across Asia and Europe that read that book and simply didn't care? Flueggea suffruticosa. In 2026, scientists at the University of York discovered something that rewrote what we thought we knew about the boundaries between life itself. This plant didn't follow the rules of evolution. It hacked them. To understand what this plant did, you first need to understand what it produces. Inside the branches, roots, and leaves of Flueggea suffruticosa, there's a molecule called securinine. And securinine is strange. It's an alkaloid, one of those nitrogen-based molecules that plants use to defend themselves against herbivores, fungi, insects. Morphine is an alkaloid. Caffeine is an alkaloid. Nicotine too. But securinine behaves differently. In modern laboratories, it baffled researchers for decades. Studies show it crosses the blood-brain barrier. It stimulates the nervous system. There's evidence of anti-tumoral activity, neuroprotective properties, and even potential for treating leukemia and neurodegenerative diseases. The problem? Nobody could figure out how the plant was making it. Every time scientists tried to map the biochemical pathway, the step-by-step process that turns simple compounds into this complex molecule, they hit a wall. The pathway didn't match any known route in plant chemistry. It was like finding a lock without knowing what kind of key exists to open it. At York, in England, Dr. Benjamin Lichman and his team decided to go deep. They didn't just want to know what the plant was producing. They wanted to know how. So they did something few laboratories had attempted before. They went looking for the enzyme responsible, the gene that codes for the first step in securinine production. And when they found that gene, the room went quiet. Because what appeared on the screen didn't look like a plant gene at all. The enzymatic key responsible for manufacturing securinine looked far more like genes found in bacteria than in plants. Dr. Lichman described the feeling. Plants and bacteria are really different life forms. It was a genuine surprise to see that this really significant plant compound was being produced by a gene of bacterial origin. But wait, how did a bacterial gene end up inside a plant? This is where the story becomes an evolutionary thriller. There's a process called horizontal gene transfer, or HGT. Normally, when we think about heredity, we think vertical, parent to child, generation to generation. DNA passed down through the tree of life. Horizontal transfer is different. It's when one organism copies genetic material from another organism that isn't its ancestor. Bacteria do this constantly among themselves. It's one of the main reasons antibiotic resistance spreads so fast. One bacterium learns to survive, then passes that information sideways to others, like a biological flash drive. But between bacteria and plants, completely different kingdoms? Science said it doesn't happen. Not in any functional, adaptive way. Flueggea disagrees. At some point, possibly millions of years ago, when this plant's roots were submerged in soil, in close contact with subsurface microbes, it copied. It adopted an evolutionary trick, repurposing chemical processing tools commonly found in microbes to defend itself. And it did more than copy. It integrated that bacterial gene into its own genome. It began using it to synthesize securinine through a biochemical route completely unlike any other known plant. It didn't just steal the code, it ran it. And when the York researchers realized what they had found, they started searching other plants. Recognizing this new chemical pathway allowed the team to find similar genes hidden in the DNA of many other plants. bacterial genes, in common plants, producing compounds that laboratories are still struggling to fully understand. What does that mean? It means that for decades, maybe centuries, we've been searching for cures in plants using the wrong map. Let's pause here, because what I just described isn't simply a beautiful scientific curiosity. It's a fundamental shift in how we understand life itself. If plants can incorporate genetic machinery from bacteria and use that machinery to create complex chemical compounds with medicinal properties that modern laboratories can barely replicate, then how many other plants have done the same thing? How many extraordinary compounds are hiding in shrubs we walk past every single day? And more disturbingly, how many of those compounds are we destroying before we ever discover them? The medical question is immediate. The discovery means scientists now have a new way to search for useful natural chemicals and new tools to manufacture them. Instead of extracting compounds from rare plants or relying on aggressive industrial processes, these genes could potentially be used to produce the compounds directly in a lab. But the deeper question is philosophical. Science spent decades classifying horizontal gene transfer as a bacterial phenomenon, something that happened between primitive organisms. Plants were seen as chemically inventive, yes, but within their own limits, within their own kingdom's borders. Flueggea crossed that border, quietly, efficiently, without asking permission, and it wasn't alone. There are documented cases of plants carrying genes from viruses integrated into their genome, cases of gene transfer from parasitic plants into host plants, cases of algal mitochondrial genes appearing in land plants. Life, apparently, does not respect the organizational charts we draw for it. It does what works. And what works, sometimes, is stealing the best tools from the neighbor next door. Let's go back to that textbook. Plants and bacteria do not exchange genetic tools. There's a curious arrogance in that, not in the statement itself, which was based on the best available evidence at the time, but in the certainty with which we wrote it, as if life owed obedience to our categories. We named the kingdoms. We drew the phylogenetic trees. We decided which crossings were possible and which were impossible. And Flueggea suffruticosa, a modest shrub with small leaves, roots sunk in the earth, simply didn't know that. For millions of years, it has been solving survival problems with whatever tools were available. No laboratories, no research funding, no peer review. It found a bacterial gene that served its purpose and incorporated it into its very being, and produced a molecule that today attracts researchers from around the world, because it may hold the key to treating cancers, neurological diseases, perhaps others we don't yet fully understand. How much time have we spent searching for cures in laboratories when the forests and the gardens and the weeds in the sidewalk cracks have already been carrying solutions that took ages to evolve? The question isn't, what do plants know that we don't? The question is, are we finally willing to listen? Nature never made the mistake of thinking it knows everything. If you made it this far, thank you for exploring the living archive with us. The discovery of Flueggea suffruticosa was published in 2026 in the journal New Phytologist by researchers at the University of York, led by Dr. Benjamin Lichman. The link to the original study is in the description below. In the coming weeks, we'll be exploring other cases where plants crossed boundaries that science said were uncrossable. Because this one, believe me, was not the first. And it certainly won't be the last. If this video made you look at any plant differently today, mission accomplished. And speaking of hidden knowledge that's been right in front of us all along, there's a book sitting on my desk right now that gave me the same feeling this research did. It's about foods. Specifically, foods that people throughout history used to survive the unthinkable. Sieges, famines, grid collapses, wars, using ingredients most of us walk past every single day without a second thought. Not survival gimmicks, not freeze-dried powder in a foil bag. Real, forgotten knowledge. The kind that kept entire civilizations fed when everything else fell apart. The kind of knowledge that, just like the Flueggea, was always there, waiting to be rediscovered. The link is in the description if you're curious. Some things are worth knowing before you need them.